So let's take a look at the first problem in chapter 3, 3.7. Well, the first one that we need to review for the makeup exam. Well, 3.7, and this is just taking a look at Mendel's dihybrid cross. So Mendel crossed peas having round seeds and yellow cotyledons with peas having wrinkled seeds and green cotyledons. All the F1 plants had round seeds with yellow cotyledons. Diagram this cross through the F2 generation using both the Punnett Square and forked line methods. So, okay, let's review some stuff here. So we have round seeds. So round seeds and wrinkled seeds. Let me move the mic a little closer here. Round seeds and wrinkled seeds, and yellow cotyledons and green cotyledons. Now, we have in class we've talked about these traits a lot, and we decided that, or we talked about that wrinkled was the recessive trait. So to be wrinkled, you need to have two little W alleles, and to be round, you need to have at least one big W allele. And to be green, well, you need to have two little G alleles, and to be yellow, you need to have a big G allele. And these are traits that are determined by two different genes. This is the gene for shape, and this is the gene for color. And we're looking at two different alleles for each gene. So remember, Mendel was working with true breeding lines. So that means if we crossed a plant that made, let's see, round seeds and yellow cotyledons, or that had round seeds and yellow cotyledons, so round and yellow, and we crossed that to another true breeding round and yellow, all the plants, all the progeny would be round and yellow. And it was because it's true breeding, we know it has two big W alleles and two big G alleles. Okay. So, and this is the P1 generation, and Mendel's crossing this plant to a plant that is wrinkled and green. So wrinkled, okay, so we need two little W's here, and I need to make sure to make my W's different sizes, but these are two little W's, and for green we have two little G's. Okay, so this is a true breeding plant also, so if we cross this wrinkled green plant to another wrinkled green plant, all the offspring would be wrinkled and green. So the question tells us that in the F1 generation all the plants were round and yellow. So if we didn't know anything about which trait was dominant, and which traits were dominant, which were recessive, we would know now, because wrinkled and green disappeared in the F1 generation, right? We see round and yellow, so we know round is dominant to wrinkled and yellow is dominant to green. And then Mendel crosses these, and he sees a certain ratio in the F2 generation. And we are going to, and I can tell you right now that that's going to be 9 to 3 to 3 to 1, because this is what we've talked about in class over and over again. So 9 round and yellow, 3 wrinkled yellow, 3 round and green, and 1 wrinkled and green. So this is the ratio. These are out of 16. So the question wants us to, and here they're all round and yellow, right? So the question wants us to map this out using the Punnett Square and forked line method to see why we get all round and yellow in the F1, and why we get 9 to 3 to 3 to 1 for phenotype ratio in the F2, and we'll also be able to determine the genotype ratios for both of these using the Punnett Square and forked line method. So let's take a look at how to do that. So to start, I'm going to just go up here, and we already have the genotypes for the P1 written out here because we knew the plants were true breeding. So I'm going to copy that, and I'm going to paste this down here. Okay, just like that. So now these plants are going to make gametes, right? So they're going to go through meiosis up here, and I can diagram this for you. So we have W is going to be on one chromosome. There's a W over here. There's one over here, and G is going to be on another chromosome. Because Mendel isn't looking at linkage, right? He didn't detect linkage. Most of his genes were on other chromosomes, or they were so far away on the same chromosome that they were unlinked. And we're getting a little bit ahead of ourselves because we're covering linkage now in Chapter 5, but we're just reviewing some stuff in Chapter 3. But okay, so W and G are in different chromosomes. So this is what meiosis is going to look like, and eventually, after meiosis 1 and 2, we are going to get gametes that just have a big W and a big G. So that is the only possibility for gametes from that round and yellow plant. And the same thing for this wrinkled and green plant. So we have the little W's on the chromosomes up here. These are homologous chromosomes that I'm showing you. I'm showing you the tetrads. W and G, the genes here, are on different chromosomes. And so this plant also can only make gametes that are genetically identical. So little W and little G for both gametes here that I've drawn here. So only one type of gamete. So the Punnett square for this one would be really easy, right? So we just have a little W and little G for one gamete, and then a big W and big G for the other gamete. And so they come together, and we end up with big W, big G, little g. Okay, so all the progeny have to have this genotype. And we can tell that, we know that big W is dominant to little w, so they will all be round, because big W gives you the round, or gives the plant the round phenotype, and big G gives the plant the yellow phenotype. So all progeny have this genotype, and all are round and yellow. So the forked line method is a little silly for this one, right? Because that's where you break the traits up into, you consider one trait at a time. And so if we did that, well, okay, all the plants are going to inherit a big W and little w, so that's 100%, so I'll just go 1 over 1. And all the progeny are going to inherit a big G and a little g, so that's also 1 out of 1. So if you multiply those across, you pretty much have 100% of the plants big W, little w, big G, little g. Okay, 100% are round and yellow. So we don't usually use the forked line method for the F1 generation, but if we did, well, that's how I would do it, right there. Okay, so let's take a look at the F2 generation, which is a lot more complicated. Okay, so let's take a look at the F2 generation. So what we know in the F1, all the plants have the following genotype. So if we self-cross these plants, or cross them to each other, well, they're going to make a couple different types of gametes, right, each plant. So why don't we, what would be the easiest way to do this? Well, I think what we can do is, let's see, let's just write out all the possible gametes it can make. So we can have big W with big G, so that's one. We can have big W with little g. We can have little w with big G. And we can have little w with little g. So that's 4 possible types of gametes. And the same thing for this plant over here. It's got the same genotype, so it can make the same 4 genetically distinct gametes. So what I can do now, and this would be using the Punnett Square method, take these, I'm going to paste those over here. Okay, so we need to include these 4 gametes in our Punnett Square. So these would be from, say, one plant, one parent. And I'm going to map out the other gametes up top here. So the gametes from the other plant. And they're going to be the same as these over here, because we're either just self-crossing this plant or crossing it to, a plant from the same generation with the same genotype. So big W, big G, big W, little g, little w, big G, little w, little g. Now, maybe I can use a different color for these. Okay, so now we just need to fill in these squares here. Okay. Oh, I wanted to change that color. Let's see. How about blue? Okay. Okay. So big W, big W, big G, big G. Let's see if I can do this without making a mistake. You could probably do it faster than me. You can fast forward if you know what I'm going to be doing here. I always make a mistake when I'm filling these out, so it will be a miracle if I don't make one. That's why I like the forked line method. It is, I think, a lot easier. Well, I'm a lot less likely to make a mistake with that method. Too bad I couldn't stream this live, and then you could tell me if I made a mistake. Someday I'll figure out how to do that. Okay, 2 little W's, 2 little G's. Now, let me see. What can I do here? I can actually, why don't okay, so if we have a yellow, let me see, I could actually draw I could use the colors to draw the phenotypes. So this one right here, this is going to be round and yellow, because we have at least one big W and at least one big G. So round and yellow. I can even color it in. It's kind of fun. Okay, why don't I look for all the round and yellows? We should have 9 of them, right? Because we already did the 9 to 3 to 3 to 1 ratio. Okay, another round and yellow here. Round and yellow here. Okay, I should only have 9 of these. Okay. I've already got 6. 7. 8. Perfect. 9. Okay. So 9 round in yellow, and we can see that because, you know, every offspring here, every potential offspring that had a big W and a big G was round in yellow, is round in yellow. Okay, so the next one we can do wrinkled in yellow. So here we're looking for anything that has 2 little W's and at least 1 big G. So there should be 3 of those. Okay, here's one. So how can I draw wrinkled? That's very wrinkled. Okay, so wrinkled up. 2 little W's. Okay, here we go. And right here. Well, okay, that doesn't, I don't think that's what the wrinkled seeds look like, but you know what they are. So, okay, now let's do, how about round in green? So round in green is going to be at least 2 little G's and 2 little G's and at least 1 big W. So here we got one here, and we have 2 little G. Yep, here and here, and then we have wrinkled and green, which is going to be 2 little W's and 2 little G's. Okay, so that's using the Punnett Square to get the 9:3:3:1 phenotype ratio, and we could even at this point go through and count up the number of different specific genotypes. So we'd have one right here for 2 big W's, 2 big G's. Well, we have one of those so far. How about 2 big W's, 1 big G, 1 little G? This is a pain doing this, but let's see if I can do it. So I'm started already. Okay, so I'll just put a little X here if I already counted that. Big W, little W, 2 big G's. Haven't done that one yet, so I got one of those. Big W, big G, little G. Haven't done that yet. 2 big W's, big G, little G. Oh, okay, we got that already right here. So I'm just going to add a little mark right there for that one. 2 big W's, 2 little G's. And I might run into trouble here because my W's are kind of looking like the same size, so I might get confused. Okay, heterozygous here for both, and I think we have one of those already. So this would be the second one. I forgot to mark it last time. Big W, 2 little G's. We don't have that one yet. Big W, little W, 2 big G's. Okay, I see that one right here. Okay, big W, big G, little G. We have that one. So let's add another one there. 2 little W's, 2 big G's. We don't have that one yet. I'm running out of space. Oh no. 2 little W's, big G, little G. We don't have that one yet. Let me go down a little bit here. Okay, we have this one already. I'm going to add that right there. Big W, 2 little G's. This one right here. 2 little W's, big G, little G. This one right here. And 2 little W's, 2 little G's. We don't have that one right here. So our genotype ratio is going to be, you know, 1 of these, 2 of these, 2 of these, 4 of these, 1, 2, 1, 2, 1. And these are all out of 16. So if we did this right, they should all those numbers should add up to 16. So 1, 2, 3, 4, 5, 9, 10, 11, 12, 13. scroll down a little bit, 14, 15, 16. Okay, so we have our genotype ratio here. So that's how we would use the Punnett Square method to get the phenotypic and genotypic ratios for the F2 generation. So what if we wanted to use the forked line method? What would we do? So I think this one's easier. So let's take a look. Now let's go back to the original F1 generation, where we are crossing the big W, big G, little g, self-crossing, or crossing it to another plant from the same generation, same genotype. Okay, what do we do? Well, with the forked line method, we separate the genes, take one at a time. So we do little Punnett squares. So even you got to do at least one Punnett square with the forked line method. At least I do. So this plant right here, this parent, is going to make two types of gametes with respect to W, right? It's going to make one with a big W and one with a little w. So I'm going to put those up here. And this plant over here, same thing. It's going to make two genetically distinct types of gametes, pollen or eggs, with either a big W or a little w. Okay, so this is our Punnett square. This is how we set it up. And we end up with two big W's, big W, big W, little w, and two little w's for a 1 out of 4, two big W's, 2 out of 4, big W, little w, and 1 out of 4, little w, little w. And you can see that because we're using a big W and a little w, and we're getting this ratio, since we're using a big G and a little g, well, we're just going to get the same ratio. So we could just, if we did that same method for G, well, we'd have 1/4 big G, 2/4 big G, little g, 1/4 two little g's. Okay, so we have these ratios. Now we can use the forked line method to combine these together. So imagine this was not a dihybrid cross, where we're looking at where Mendel and the P1 crossed two plants that are different for two traits there. Imagine we were just looking at one, just looking at one trait. So we've already seen that if we were just looking at, round or wrinkled, 1/4 of the progeny would have two big W's. 1/2, or 2 out of 4, would have a big W and a little w. And I'm going to move down a little bit here. And 1 out of 4 would have two little w's. So that would be just the genotypic ratio if we were just looking at one trait. So of all the offspring, 100% of the offspring, 1/4 of them are going to have this genotype here, 2/4 are going to have this genotype, and 1/4 are going to have this genotype down here. So, but we're not looking at just one trait, right? So we're looking at another gene. We're looking at round in yellow and green, and we've already calculated. We've already know that 1/4 of the plants are going to have these two big G alleles. 2/4 are going to have a big G and a little g, and 1/4 are going to have two little g's. So we can use the forked line method because we have that ratio for the G alleles. Well, now we know that 1/4 of these plants with two big W alleles are going to have two big G alleles, and 2/4 are going to have a big G and a little g, and 1/4 are going to have two little g's. So the same thing of all the plants that have a big W and a little w allele, 1/4 of those plants are going to have two big G's, 2/4 are going to have a big G and a little g, and 1/4 are going to have 2 little g's. You know, so we know the ratio of all the plants are going to be 1 to 2 to 1. So we're just kind of breaking it down and grouping the plants into plants that have 2 big W's, and of those, you know, we're going to see the 1 to 2 ratio, 1 to 2 to 1 for the G's. And the same thing with the half of the plants that have a big W and a little w. Well, we're going to see, if we just look at those, we're going to see a 1 to 2 to 1 for the G alleles. So let's look. Same thing for these down here. 1/4, 2 big G's. 2/4, big G, little g. And 1/4, 2 little g's. And this is this concept right here is a little. It's sort of the next step. Is that this happens because these alleles here are the alleles for W and G, or the alleles for the color gene and the shape gene, are segregating independently. So we're seeing independent assortment here, and this doesn't really work when you're doing linkage. We have to do something a little bit different. But because these are sorting independently, we can just use this forked line method with these ratios like this. So we would just multiply these across. So that's 1/4 times 1/4. It's 1 out of 16. One quarter times 2/4 is 2 out of 16. And then these are 2 big W's, big G, little g. This is going to be 1 out of 16, 2 big W's, little g, and 2 out of 16, big W, little w, 2 big G's, 4 out of 16, and then 2 out of 16, and let's see, 1 out of 16, little w's, big G's. There's a little pattern going on here. So 2 little w's, big G, little g, 1 out of 16, 2 little w's, 2 little g's. Okay, so let's take a look at these phenotypes here. So 1 out of 16 are going to be, I want to put the colors in here, yellow and round. 2 out of 16 also going to be yellow and round. And these right here are going to be round and green. Yes. Okay, let's take a look down here. 2 out of 16, what do we have for there? Yellow and round. Yellow and round again. And for this one we have round and green. Okay, and all these down here are going to be wrinkled. We're going to have wrinkled and yellow. We are going to have wrinkled and yellow again. And then we're going to have wrinkled and green right here. Okay, so is this a 9 to 3 to 3 to 1 phenotypic ratio? We already have the genotypic ratio, right? We got all those right there. Okay, that's the genotypic ratio. This 1 to 2 to 1, 2 to 4 to 2, 1 to 2 to 1 here. But let's see if we have 9 yellow and round. We have 1 here, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so we have 9 yellow and round, and we have, let's see if we have 3 green and round. We have 1 here, and 2 here, so that's 3. Let's see if we have 3 yellow and wrinkled. We have 1 here, 2 here. Yeah, okay, and let's see if we have 1 green and wrinkled, and we do. Okay. So that's it. Problem 3.7 is done. Kind of a long problem, But if you understand that, then you understand Mendel's dihybrid crosses and how to solve them by the Punnett Square or the forked line method.